Yeah, I want to thank the organizers for inviting me to this stimulating meeting. I also want to thank Roger for introducing many of the genes that I'm going to talk about in the next 30 minutes or so. So in my group, we are interested in how cell fate choices are executed. The model that we use is blood development in the embryo and also blood development in the adult homeostatic blood system. I'm going to talk about those three issues today. Our investigations of the diversification of early mesoderm in the mouse embryo, and then on two other stories that are to do with regulatory network models in the adult hemopoietic system. And in all of this, we are wanting to learn how networks of transcription factors play a role in making those cell fate choices, and also want to pay attention to the fact that those cell fate choice decisions are made at the level of the individual cells, so that we actually need to do quite a lot of single cell analysis in order to move us forward. So then to the first story, which is about early mesoderm diversification. And what we've done here is that we have surveyed at single cell expression the emergence of mesoderm in the mouse embryo. And it's a total of 1,200 cells that we've analyzed here. And it's from this time course, day 6.5 to day 8. And these are embryos here that we have dissected, and the cartoons to the right, they show the cells that we have profiled. So from the day 6.5, we profiled the distal portion of the embryo, which is largely epiblast cells. Then from day 7, 7.5, 7.75, based on those anatomical staging criteria, we isolated FLIG1 positive cells, FLIG1 positive cells, and CD41 positive cells, and then again at the later stages, FLIG1 and CD41 positive cells. So FLIG1, Roger already introduced. This is a marker in our case for all the mesodermal, I have to say it the other way around, all the cells that have potential to turn into blood cells in a mesoderm, they will be expressing FLIG1. And then once they have actually arrived at being blood cells, they will express CD41. So that's where this sorting strategy comes from. So first thing you might want to do is to do clustering. You saw some of this morning. So that's just to see, well, what types of cells are in our data set. And there were 10 major clusters they're shown here, just with some sort of key marker genes. For example, cluster 3 here, that's the epiblast cells. And you can see that they express OCT4 and non-OCT. There are other clusters, such as this cluster 8 here. That's primitive red blood cells, the cells that Roger talked about. They express very high levels of embryonic globin. These are their precursors, cluster number 7. They already express TAL1 or SCL, the gene Roger talked about, but they specifically then express GATA1. We also have pharyngeal mesoderm that we heard about yesterday. That's this cluster here, or allantois is cluster 9. These are all then here specifically genes highlighting those, which is telling us that FLIG1 isn't just picking out mesodermal cells that are destined to go to blood, but also to some other mesodermal tissues. Clustering really only gets you that far, so you need to deal with this multidimensional data in some other ways that can take account of how might cells be related to each other. So this is dimensionality reduction. In our case, it's a TSNE plot. So this is the 1,200 cells. In this diagram, if cells are similar to each other in all the dimensions, and in this case it's 2,500 because that's the number of genes that were heterogeneously expressed above technical noise in our data set. So we've reduced those 2,500 dimensions down to 2 dimensions, but what's maintained is if two cells are similar in the high dimensional space, then they're near each other in the low dimensional space. And you can see that there's some sort of groupings emerging here. They're colored in based on the day when we collected the cells, so the turquoise is the day 6.5, and then it goes purple, green, red for the 7, 7.5, and 7.75. And here, it's the exact same diagram, but retrospectively colored in based on the clustering that we've done. So here you can see that big cloud here. That's the epiblast cells. This is the primitive red blood cells, their precursor. This red cluster here will become important. These are the endothelial cells that Roger talked about that give rise to the second wave of yolk-sac hemopoiesis. So that allows us to visualize, in a way, early mesoderm diversification. So I'm going to talk a little bit about this epiblast cloud. So this is the epiblast cloud. They're not all the same. There's some structure in this cloud. And that's shown here if we plot the expression of brachyury just in this cluster number 3. So you can see that down here there is then a group of cells that turns on brachyury, and this is not just random. We can color in some other genes that are known to be associated with gastrulation, and then you can see that, for instance, MESP1 comes on even a little bit later than brachyury, etc. So that it appears as if we can recover cells at different stages of mesoderm induction, basing this on expression of these types of genes. We can take this a bit further because we can just now rank all of these cells. These are about 500 epiblast cells that we have, based on their level of brachyury expression, and then we can go into the whole transcriptome and say what genes show positive correlation or negative correlation with brachyury, and then we get known, but also potentially novel genes that might be implicated in gastrulation. So that's a platform to discover potential new players in the process of mass gastrulation. Now I'm going to talk a little bit about this blue cluster here. So these are FLIC1 positive cells that are largely from the early 7 and 7.5 stages when we sorted FLIC1 positive cells. Based on genes that they express, we labeled them as nascent mesoderm, and what we mean by that is that they're most likely cells that are only just coming out of the primitive streak and when they first turn on FLIC1. So that's, again, this cluster here. Bioinformatically, we were able to identify that it's this dimension here that has the biologically meaningful variation in this cloud. And then we tried to figure out, well, what's going on here? There were clearly genes were differentially expressed when you went along this axis. So for a long time, we thought, oh, we can do something a bit similar to what we heard about this morning. Is this a differentiation time course that we're dealing with? But it turned out that it's space. So genes that are known to be expressed more on the anterior side, they were high on the left, low on the right. And genes that are known to be expressed more highly on the posterior side, they were high on the right and low on the left. So what it actually seems to be that in this particular case, it's not pseudo-time, it's pseudo-space that we can identify bioinformatically from this data set. And then once we've done this, we can then, again, go back into the entire transcriptome and find genes that would cluster along this pseudo-space axis and would be high on the left, high anterior, low posterior, or the other way around. And then there was a third cluster that was high at both ends, but lower in the middle. So again, it's kind of a platform to discover new players. I want to quickly talk a little bit about this cloud here. So in this case, these are the precursors and then the more mature primitive erythroid cells. In this case, we believe it's actually time that's recovered, because you can also see that the yellow cluster is mostly purple and green, and then when you move into the brown, it's green and red, and this represents the actual time of when the cells were collected. So here I think we are dealing with pseudo-time. We can do the same as I've just talked about. We can now line up all the cells based on this axis and then identify genes that are downregulated or genes that are upregulated. And there's also genes that are upregulated and then go back down. And it contains a lot of the known players. This is the embryonic globin that goes up. This is one of the genes that I think was in one of Roger's diagrams. It might have been SOX17, but this is one of the genes. We know if you overexpress this gene in mouse embryos, you stop this process. So this downregulation isn't incidental, it's important. There's also this gene here, GATA1, Roger talked about. This is kind of the major regulator that drives this erythroid differentiation. We therefore also performed some complementary GATA1 ChIP-seq data, and were able to show that in particular the upregulated genes were associated with binding events for this factor. Okay, so I just want to take this a little bit further. If we think about early mesodermal diversification, there's really two ways you can think about it, and I think we kind of touched on this a little bit in the meeting. Is this a stepwise process of fate restriction, where you start with, let's say, an epiblast cell, then it turns into a mesoderm cell, and then step by step it has to lose potential. It has to lose cardiac, then it has to lose smooth muscle, then it loses endothelial, and in the end you're left with a blood cell. So that's intuitive, and in fact a lot of people who want to produce mesodermal derivatives out of embryonic stem cells, they kind of try to work on this framework. But a lot of the classical lineage tracing and also modern lineage tracing using cremite actually suggests that it may be more like this, that it's independent fate acquisition, that you're starting, that it's more or less journeys that have a known endpoint right from the start, from when epiblast cells get specified to be mesoderm cells. We can't really make any contribution to these sorts of important debates just by profiling single cells in an unperturbed system. So what we've therefore done is moved on to a perturbation. And that's the perturbation. This is the gene that Roger talked about, TAL1 SEL. Here I'm listing why it's really important in this early mesoderm diversification. In the mouse, Roger's group and some other groups have shown corresponding data in vertebrate model systems, zebrafish and xenopus. What Roger didn't mention is that there's also this paper from 2012, which suggests that when you lose TAL1, and therefore the journey towards blood is blocked, that there is an ectopic fate that these cells will then move into, and that's cardiomyocyte, and that therefore TAL1 is a gatekeeper to a binary fate choice. But because in the early embryo, the cell numbers are so extremely limiting, the molecular defect that gives rise to those phenotypes is actually not known. So this is an in situ hybridization showing the expression of TAL1. That's the yolk sac staining. This is more the intra-embryonic staining. This is the expression in our single-cell data. So you can see it's obviously it's in the right place. These are the primitive red blood cells. This is endothelial cluster there. Mouse knockouts. We analyzed the mouse knockouts. In the knockouts, you get production of FLIC1 positive cells, but they cannot turn on CD41, whereas the wild types can. So that's what I said. They cannot move beyond this sort of endothelial stage. So what we've done then is profiled by single-cell RNA-seq cells from wild-type littermates and TAL1 knockout new embryos. And then we projected those new data onto the reference map that we had generated before. And this is the new data. The wild types are the purple triangles, and the knockouts are the light blue ones. First thing we can see is that then only wild types project onto the yellow and brown cluster. So the knockout doesn't produce any of this primitive lineage. And we also can't, from this analysis, detect where this differentiation might be blocked. So this must happen really, really early, earlier than we looked. But of interest, this endothelial cloud, which is the hemogenic endothelium, that we can project both wild-type and knockout cells onto that space. So what we then did next is to do differential gene expression analysis of wild-type and knockout cells for this endothelial cluster here. And then we also interrogated to what extent the differentially expressed genes were related to the blood program. And the way we defined the blood program is by comparing this nascent mesoderm to all of those single cells that we have that are associated with blood production. And what we see here then, this is the 50 most downregulated genes in the TAL1 knockout endothelium. Half of them are associated with this blood program defined in this way, and there is another half that are also downregulated. What this plot shows here is the actual expression differences, and I'm going to get to that in a second. So what we see is we see downregulation of the expected blood program. We also see downregulation of genes that are currently becoming very interesting, like this gene HOXB5. There was just a paper in Nature to say it's a very specific marker for definitive blood stem cells. Single-cell resolution, I think, reveals these gene-specific patterns, which I think can give us clues about regulation. So, for example, what this plot shows here, this is the level of expression, and this is the frequency with which we have seen it in single cells. And the black curves are the wild-type cells, and the blue curves are the knockout cells. So if you look at SOC7 here, you can see it's more or less unimodally expressed in the wild-type cells. In the knockout cells, it becomes bimodal. Some of the cells express, and some, this is near the detection limit, so they don't express. Another gene, HHEX, more or less remains unimodal, but has a quantitative shift. So those sorts of differences you would not be able to see if you did a conventional RNA-seq experiment. And I think it may well have something to do with what are the partner proteins that TAR1 might be cooperating with in order to control expression of SOC7 versus H hex. So I think there's an opportunity to investigate mechanisms. But what was surprising is that we saw no signature of upregulation of the cardiac program, even though it had been suggested that this is what should be happening. And this is what had been suggested in this paper in Cell. And what we did then is they had defined gene sets that were associated with the blood program and the cardiac program. And what we could see is that all the genes that they had associated with the blood program, they were downregulated. And what's in red are the genes that are statistically, highly statistically significant in our data. But the genes that they have labeled as being associated with the cardiac program, there's really no significant changes here. And what's important to bear in mind is that their expression analysis was done 1.5 to 2 days later than we were looking, because at the very early stages, there's so few cells that you cannot really look at it unless you do the single cell analysis. So there's no early upregulation of cardiac genes in TAR1 endothelial cells. And I think what that means is that the model that they put forward, that this is a binary gatekeeper that sits at one of those decision points, is unlikely to be correct. And that it is more likely to be an executor of one of those developmental programs. And when you don't have it, we probably get a process of transcriptional reprogramming or transdifferentiation. So if you think about this in the context of a Waddington landscape, is the action here or here? And I think it might be here, but if you knock it out and you can't move further, then the cells sit here. They're susceptible to receiving signals that might then push them over this ridge or push them backwards so that they then can roll down a different valley. And this is, incidentally, this is much more consistent with the older embryology, the classical embryology data, because we know that epiblast cells that give rise to endothelium and blood cells, they are different. They are in different locations in the epiblast. So that in reality, a cell is not ever being asked to make this fate choice. But instead, it really matters when and where they enter into the primitive streak, because they are immediately programmed to go down a given journey, and unless something drastic happens, that's the journey that they're on. And I think there's a danger here that what we thought was these clear-cut molecular data of knockout experiments that are actually maybe a bit more what I would say is we need to pay really good attention to the classical experiments that nowadays might be labeled as descriptive, but if they're describing the real system in an unperturbed way, I think that tells us what is the kind of baseline that our interpretation needs to be consistent with. So that's the conclusions. I don't think I need to go through that. I just want to mention here that the scale and complexity of all of this is really quite enormous. We're doing this in collaboration with John Marioni from the EBI. Okay, so now quickly on to this part. This is really inspired by Eric Davidson. And it's a network model that we have built for the adult hemopoietic system. Roger had a similar map. And you can think of this as a developmental system because there are these stem cells, they give rise to those mature cells, and they do have to make cell fate choices during this process. There's one big difference, and this is that the stem and also some of the progenitor states, they are stable. So we just touched on this in the discussion of the last talk. Because in those embryonic systems, let's say epiblast cells, they only exist for 2 days. There's never, ever a cell of that transcriptional state around. But here, these cells, they have to last us a lifetime. So what we've done here is to carry out a construction of a network model bottom up. And this is why I said inspired by Eric, where the idea is that the genes and the gene regulatory elements, they are all physical entities. We ought to be able to describe them, functionally map them, map their activities. And that's what we've done, build a gene regulatory network model bottom up by studying the cis regulatory elements and the transcription factors that interact with them. The transcription factor interaction, so this is a 9 transcription factor model. We generated those by carrying out chip-seq experiments in 2 model cell lines. So that tells us where in the gene loci those factors are bound. Then we tested all of the regions that were bound by 2 or more factors and that show active histone marks in transgenic mice, because we wanted to validate them as all as true regulatory elements. And that gave us a total, well, for this study, quite a few of them are known, but we identified 9 new ones. And that's the sort of assay that we do. We fuse a piece of DNA to a LAC-Z reporter gene, make transgenic mice, and then test whether or not it shows activity in the blood-forming tissues. In this case, the fetal liver and the dorsal aorta. So this is a dorsal aorta picture here. And these are the clusters that Roger referred to. And then lastly, we wanted to know, well, now that we know where are the elements and what binds to the elements, what's the effect of the binding? And the way we measure this is by generating mutant constructs of all of those enhancer and promoters that fulfilled these criteria for the motifs that correspond to the factors that were bound to them. And that was a total of over 1,000 mutation assays that we had to do. But what it showed us is that for all of the 9 transcription factors, we saw that they could either be activating or repressing. So they are kind of, some of them are almost always repressors or almost always activators, but there wasn't a clear-cut rule. So if we just went by binding and said, oh, this factor is meant to be an activator, it's activating, that would have been dangerous. So that's the model that comes out of it. So it's a little bit different to the models you saw before, because we actually pay a lot of attention to the regulatory elements, because that's where we have a lot of the functional data. But they then drive the genes, and then we know the genes will produce a protein that will then feed back onto the other regulatory elements. So we then went on to use this experimentally validated network model to simulate transcriptional networks in single cells and validated predictions by single cell expression profiling. We built a dynamic Bayesian model, a 3-tier model here showing the motifs. Then a group of motifs makes up a regulatory region, and a number of regulatory regions. They will determine the activity of a gene, and then the activity of a gene determines the probability of a motif to be bound, and so on. So we can set a starting level of genes, and then we can execute this model and run it over time. So that's what we've done here by starting all of the genes at a midpoint and then run the model. And that's what it looks like, that many of the, or some of the transcription factors, they stabilize at a given level. Some others, they oscillate around a characteristic midpoint. Importantly, the model allows for simultaneous expression of all of the 9 factors, which was important to us because we know that that's the case. We can also visualize the data differently, where we run 1,000 simulations, and then we show the same plots I talked about before, which is the level of expression against the fraction of cells that exist at a given level, and then we see those different patterns here. And the reason this is a useful representation is because we want to compare this to real single cells, and we can't follow real single cells over time for 9 factors, but we can do lots of snapshot measurements on real single cells and then compare those single cell snapshots to the single cell simulations. So what we then went on to do is to perform network perturbations, and that's shown here, where the wild type is the dotted line, and here we knock down Q1, the factor that Roger talked about. And it's really easy to knock down things in a computer model. So that's here. But what happened in the computer model is that none of the other genes really changed when we knocked down Q1. But when we did the experimental confirmation, Q1, actually, we have a pretty good construct. So we can virtually knock out Q1 in a significant proportion. And because this is QR to PCR, this is a log2 scale. Even here, this is quite a significant drop. But interestingly, only one of the other genes actually changed its expression level, and that's this gene here, RUNX1. So the expression state is relatively stable, both in the computer and also experimentally. And we saw this for 2 other single factor perturbations. If we do double factor perturbations in the model, then it's much more fragile that it can fall apart. But they are harder to do experimentally. But we did one further experimental sort of validation experiment, and this is that we simulated activity of this fusion protein that's common in patients that have leukemia. And there, this gene, RUNX1, is also known as AML1 because that stands for acute myeloid leukemia 1. It's fused to this protein ETO, and that is thought to create a dominant negative repressive version. So what we've done, therefore, in our computer model is to convert every binding event of RUNX1 into a repressive event. And then ask, well, what does that do to the model? And now there's a bit more happening. For example, we see here changes in PU1, we see GFR1b, PU1, etc. This is relatively easy to do experimentally, and that's what we've done here. Now we also see in the experiment quite a lot of changes, and some of them are the identical changes, like here, loss of GFR1b and loss of PU1. So they're major expression changes. I'm just going to talk about the bottom one here. I think having this model is not going to be correct, but I think it's useful, and it lets us think about potential mechanisms at play. And we've done this work together with Marella de Bruyne and Nicola Bonzani. Okay, so very quickly on the last point. So what we've done here is, on this hierarchy of hemopoiesis that Roger and I both have talked about, these are different populations that we can purify single cells, and we've profiled single cells, this time by quantitative real-time PCR, measuring 48 genes. In total, 2,500 genes. This is how you can plot those. So that's, again, one of those dimensionality reduction approaches. All of the dots are the cells. I've now colored in retrospectively. These are the stem cells, and these are two of the directions, the ones, in fact, that Roger was talking about, the red blood cell and the myeloid directions. So you can think of these as snapshot measurements that kind of represent the transcriptional landscape, and therefore differentiation trajectories. So we define differentiation trajectories in this direction, the myeloid direction and the erythroid direction here. We can then, again, line up the cells in pseudotime, and then we can see upregulation in this case here of Notch, downregulation of GATA2. This is the other trajectory. I don't really need to talk about it. What we've done then is to try and use the pseudotime ordering to infer a Boolean regulatory network model, where we have calculated correlations between all pairs of genes to find the most highly positively or negatively correlated gene pairs. And then we've used those as possible functions to investigate the time course, where we would basically say, out of those functions, A activates B. Does that make sense? If B gets induced here, is A expressed there? So then we have found those functions that best match pairs of cells when we move across this time course. And these are the two networks that we get out of this. And in fact, they are actually a lot more complicated than this, because here we're just drawing links. But most of these links are complex 2 or 3 factor Boolean rules. A plus B, not C, does something to D. So what we've done next then is to perform stable state analysis, to just ask, where can these two different networks stabilize? And that looks pretty good, because the myeloid network has stabilization points down this trajectory, and the MEP megakaryocyte erythroid stabilization points down this trajectory. So very last thing then is we wanted to do a little bit of validation here. There's still some work ongoing. For this, we turn to 2 cell lines. This is a cell line that was meant to represent this megakaryocyte erythroid direction, and this cell line is meant to characterize this lymphoid myeloid trajectory. In order to satisfy ourselves that this might be useful models, we did some single cell expression profiling of those 2 cell lines, and it was good for us that this cell line here projects down there, this cell line projects here, so they seem to be relevant, exist in relevant transcriptional states. What we've then done is compared the 2 networks and identified rules that are only in one but not the other network. In this case, GATA2 regulating those 2 genes was only in the MEP network. We've then done chip sequencing for a number of factors, a total of 10 factors, in those 2 model cell lines, including this factor here, GATA2, where we can see it has a lot of binding in this CBFA2T3, also known as E22 gene locus, and there's very little GATA binding in the cell line that corresponds to this network. We then tested this promoter, both a longer and a shorter version, wild type, and mutating the GATA motif, and that went down. And then we've done the same for the NFE2 promoter. Again, very strong binding in one, but not the other condition. If we mutate the Gatta motif, the activity goes down. So binding and activity are both consistent with these network models, with those links existing in this one, but not in that one. So again, I hope I have explained this. We've done this together in collaboration with David Kent, who is our source for all advice in terms of cell sorting. Okay, so in summary then, there's a lot of interest, I think, in single-cell genomics to study rare cell populations, cancer or stem cells, resolve heterogeneity, investigate clonality in cancer, but I think there's also tremendous opportunities to learn something about developmental trajectories and also to consider regulatory network reconstruction and thereby gain mechanistic insights into those regulatory processes. And I want to acknowledge my group. These are key people who contributed to the work that I talked about today. These are collaborators that were key to what I talked about today, and these are the funding agencies that fund our work. Thank you.